Good morning, everybody. We're going to go ahead and get started here. I'm going to just a very quick announcements. This evening is the Women's Networking event, and if you don't already have a ticket, there are still opportunities to obtain a ticket. And I think there'll be at least one group walking from the registration desk at about 6 o'clock tonight if anybody wants to join us for that. So without further ado, I'm going to introduce a member of our local organizing committee, Professor Roel Vermeulen. Trying. He's going to tell us about our plenary speaker this morning. Thank you.
Thank you, Lisa. Welcome everybody in this early day on Tuesday. I really appreciate it because if you didn't realize, you had an option this morning. We're actually gonna live webcast the whole morning, so you could have stayed in the comfort of your hotel and switch on your laptop and watch from there. But I'm very happy that you all showed up this morning. I'm going to introduce Chris very shortly. I just wanted to give an introduction to what we're actually trying to do today. We have a symposium on the Exposome during the whole day, in which we actually want to address exactly the topic that Chris is going to talk about. How do we move from a concept, an idea, to actually put that into practice? And I'm very happy to have Chris Wild here, but also to have most of the senior investigators involved in the large exposomics projects around the world, from Japan, the US, and from Europe, to actually reflect on how we're trying to actually move this concept forward. In the afternoon, we'll actually go to some practical examples of how that's done, to end up in the late afternoon to talk about how do we actually come to statistical and causal inference. How do we actually know what is real and what is not real? So I hope you will stay for the whole day. But again, it will be webcasted. It will actually be recorded. So there is a chance to actually look it back when you're at home. It's a real pleasure to introduce Chris Wild today, who's going to give a keynote. And Chris is really the person that coined the term in 2005 and is really a leader in this field and has fostered this process forward for the last 10 years. And so it's a real honor to have Chris here and actually giving this opening address to really reflect on the conception of the idea, to actually how it's actually moving forward and how the IARC is also thinking about moving this forward. Chris Wild is now the director of IARC. I think since Chris started at IARC, there has been a large change at making the IARC even better than it was, and really giving a new impulse to this kind of research. Chris had, after he did his studies, a very short stay in the Netherlands, as I saw, so I'm hoping that he actually is enjoying being back into the country where he did his postdoc. Then he had a professorship in the University of Leeds, mostly focusing on biomarkers for aflatoxin and other exposures in relationship to liver cancer and esophageal cancer. Chris is still very active in research, and I think is one of the leaders, and I'm very happy that he's here. So Chris, the floor is all yours, and looking forward to it.
Okay, well, thank you very much, Roel. And it's one of the few places in the world where I have to push the microphone down when I get up to speak. As Roel said, I spend a year here, and it's the only year in my life where I spent 2/3 of the time looking upwards when speaking to people. So it's a great pleasure to be back here, and thank you for this opportunity. In preparing for this presentation, my mind was taken back to 2009 when I attended the ISES meeting in Minnesota, St. Paul. And aside from the bitterly cold weather, remembering that my European coat was totally inadequate for purpose at that stage, I remember two things about that meeting, two things in particular that struck me. The first was that I became aware just of the depth of work and knowledge and experience that was in the field of exposure science, and that, to my own shame, I have to say, I was unaware of quite the scope and depth of that work. And the second thing that struck me was there was almost not an epidemiologist at the meeting. And I felt that was a huge opportunity there to bring those disciplines together. So when I saw that the title of the meeting, which has now gone off the screen, had the word interdisciplinary in it. I thought this is really a great topic for here, and I hope I can add some comments in that context of interdisciplinary research. So let me just see. So I'm going to split my talk into 4 parts, talking about the exposome. First of all, the prompt for it. What was the purpose behind this idea? Second, the promise. Then I'm going to give a few examples of the practice at the agency, but avoiding really what you will hear about later in the day. So a lot of work going on now, which I won't cover, of course. And then I just want to give a personal perspective at the end, some of the challenges that I think we face in this field. So first of all, the prompt for the exposome. This slide shows you the projected increases in total number of cancer cases globally, split by level of human development index from very high down to low. And this is just in a 20-year period. So something like a 60 to 70% projected increase in the total cancer burden in just a 20-year period globally. And this is driven predominantly by aging and population growth. And actually, if you look at the percent increases that the different levels of human development countries will face, you can see that the heaviest increase in burden is falling on the lowest human development index countries. And these, of course, are the countries with most difficulty to actually face that challenge. So I have this impending, what Margaret Chan, the Director General of WHO, has called a silent tsunami of non-communicable disease, including cancer. The other challenge, of course, is the spiraling cancer costs. And this is a challenge even for the most developed countries. And I picked an example here because it just recently came out in JAMA Oncology, and I think it serves the point I'm trying to make. This is showing the expenditure in the first year after a new anti-cancer drug is released, and just showing the cost here from 2000, when it was about $2,000, up to 2014, when it's just under 11,000. These are a very specific set of anti-cancer drugs for outpatients, so it's not representative of the field, but it shows this trend. And this, of course, is in the US, ranked 8th in the world on the HDI scale. If we move to India, ranked 130th, it's a different profile, but underlying is some of the same challenge. And here what you see are catastrophic out-of-pocket expenses that fall on a household. 40% of the annual household expenditure can go on inpatient cancer care. And you see then reflections, the impact that has on the family, in participation in the workforce, and so on. So huge impact of cancer treatment costs. So the conclusion we came to at the agency is really that no country can afford to treat its way out of the cancer problem. And this is not to at all say we don't need the advances in the latest cancer treatments, of course we do, but we need to complement this by much more emphasis on prevention of the disease and early detection, and have this balanced, integrated approach across the board. Cancer prevention is, I think, under-supported and under-promoted to some degree. We say around half of all cancers could be prevented by applying the knowledge that we have. But we also know, of course, that the majority of cancers have a lifestyle or environmental etiology. So the potential is even greater. We were at a meeting recently with the European Cancer Leagues, and one of the cancer leagues brought a pillbox, which said, I forget what the name was now of this product, but when you opened it, inside were 12 things you could do to reduce your cancer risk by prevention. And they were trying to make the point that if we could put in a pill form something that prevented 50% of cancers, really would be worldwide news. And I just want to make the point at the end of this slide, and sometimes forgotten, for many common cancers, despite all the epidemiological research, there are many that are still of largely unknown etiology, either globally, like prostate cancer, or regionally, cancers of the esophagus in some of the highest incidence regions of the world are still a very obscure etiology. And I put on this slide a few examples of some common cancers for which we still don't have major risk factors identified. Prostate I've mentioned already, but lymphomas, leukemias, kidney, pancreas, thyroid, brain, you could add others actually, where we know perhaps a little about the cause, but not the major causes. So to my mind, this still implies the need not only to implement the preventive measures we have, but also to continue to study the etiology of the disease. So that was some of the context really to this call to complement the genome with an exposome. The causes that I've mentioned. We know in this room certainly that exposure measurement is problematic in many areas, leading to misclassification in epidemiological studies. I felt also at the time that there was huge investment going into prospective cohort studies. Particularly I was involved in one in the UK, £65 million at the beginning, 500,000 people. And that investment was predicated on being able to measure both the environmental lifestyle exposures and the genetic risk factors to understand chronic diseases. If we couldn't measure one half of the equation, how could we move forward? And then there were some major advances both in cancer biology and technology which seemed to make the time ripe for this field. And Paul was just reminding me before that I used this image of the male fiddler crab to illustrate the exposome. Because when I saw this image and this one huge claw, which this crab has invested, I think 50–60% of its body weight in one claw to attract the female fiddler crab. And then I saw the imbalance with the other claw and thought, this is where the funding has gone in the last years in terms of genomics versus the ability to interrogate the exposures, environmental lifestyle exposures, and this imbalance is not helping us. And in fact, this crab cannot feed itself with the big claw, relies on the small one to get something into its, the food into its mouth. So the definition, this is taken from the Dictionary of Epidemiology, 2014, Michael Porter. And I like this definition because it alludes also to the purpose of the exposome. A potential measure of the effects of life course exposures on health and the totality of exposures to which an individual is subjected from conception to death. And as I go through my presentation, I'd like to keep in mind the purpose of what we're trying to measure. And I keep this slide very close to me actually throughout my own research. Because and a lot of my own research has been in Africa, and there you really want to see the science translated through to some change that reduces disease burden. So not only to count, but also to effect change. That's the purpose of epidemiological research in particular. So what's the promise of the exposome? I think first of all, this paradigm shift to capture a far greater breadth of exposures for a given person. And that may translate to these semi or agnostic approaches to generate new hypotheses. But also I think there's an important opportunity to consider for the first time some of the complexity of mixtures, co-occurrences, interactions, and synergies between exposures. And of course, this second parameter of time, across the life course and the differential effects of a given exposure depending on the timing of that exposure. In trying to bring that together, this cartoon was in a paper in 2012. I thought it was interesting to think about this general external exposome, looking at social capital, education, the urban-rural environment, and so on. As well as the specific external exposures that we tended to focus on previously, and then the internal exposome, the things like endogenous hormones, body morphology, gut microflora, and so on. And that this together would constitute the human exposome. And the timing, and I'd like to come back to this point, that because I think it's one of the differences with the study of the genome, that clearly, ideally, you would want to track that evolution of exposure history throughout the lifetime of an individual. But this is not practically possible, and so we're likely to look more at specific crucial time points across a life course. So in a way, the challenges in characterizing the exposome are actually the strength of the concept itself, the scale and complexity of what we're trying to capture, stretching perhaps all the way from things like the gut microbiome through to the built environment, the dynamic nature of the exposome, the fact that those exposures will vary over time, perhaps linked to changes in occupation, major lifestyle changes, but also some technical and data analysis challenges. And these are becoming clearer as we try to put this into practice, requiring new bioinformatic and biostatistical methods. But I want to insist on the fact that even a partial characterization of the exposome can take us forward quite significantly in the field. I think it would be an error to think that the exposome is mirroring the genome. I think it complements but does not mirror. And the reason I think that is that first of all, the exposome in its pure sense for an individual, its entirety, is unlikely to be characterized. It'll be partly characterized across many individuals. So therefore, while the application of the genome to health may be at the individual level, I think the individual level application of the exposome is unlikely. And I suspect it'll be more used, as we're seeing it used, at a population level to understand the causes and the prevention of chronic diseases. And therefore, the value is likely to be found in public health benefits. I think it's a little bit broader than just understanding the causes of a disease, and I just wanted to make that point at this stage. Of course, that's a major drive for investment in this area, improve exposure assessment, capture these co-exposures and assess confounders and so on. I think it can give us some better dose response data for risk assessment. And I'll come back to this third point, the biological plausibility of associations that we see at a population level between exposure and disease. I think this can really help us with that aspect of establishing causality and bridging experimental and human data. I think some of the measures we develop may be very useful for evaluating interventions, though, as well, mechanism-based biomarkers in particular. It can help us in stratifying risk, both across a population, but also by time and over the life course. And also, as you well know, this could help us also in terms of biomonitoring surveillance approaches as well. I think it's important as we look at some examples to bear in mind the difference between the science and the technology. I mean, whether we're thinking about the questions or focusing on the tools. And I notice there is sometimes some confusion between the exposome and exposomics and the focus on the technology rather than the underlying questions and the things that we're aiming at. So the tools are broad. And although I will give some examples about the biological approaches to measuring parts of the exposome, of course, I just wanted to emphasize that there are many other exciting technologies that are being applied now to this field and are driving the field forward. Certainly GIS-based environmental modeling, for example, different mobile devices, the use of pictures to record things, as well as increasingly refined questionnaire approaches. And those together with the sort of biological approaches are improving the field. And I think here there's a huge opportunity. We're very aware, and I'm speaking now about the field I know, which is the cancer field, that the basic science has moved forward tremendously over the last 10 to 20 years. And this is finding a very natural outlet into the clinic and personalized treatments for cancer. But actually, we've I think, missed an opportunity at the moment to drive some of that knowledge and associated technology back towards the population and some of the public health challenges we face, and the causes and to study the causes and prevention of cancer. And yet, we actually are understanding that the risk factors in the environment are what drive the specific molecular changes underlying the development of cancer. And those same changes are what form the basis for the treatment strategies. So there is actually a direct link between the exposures, the molecular alterations, and the prognosis. So the same cancer from different parts of the world will actually have a different molecular profile because of the different exposures. And that really links the exposure part through to the clinic. And many of the methodologies that have been developed to interrogate the molecular profile of cancers for the clinical research can actually be applied on the sorts of biospecimens that we would collect at a population level. So I think there's a double benefit here to be had from the advances in basic science. So I was thinking back for this presentation, 2005, we had this newborn baby, the Exposome. And many of us may have experienced the teenage years of children. And we're, I guess, now approaching the teenage years for the Exposome, where we have to try and take this nice concept into the difficult world of research on human populations. And it's actually a very young field because the infancy of the Exposome was anonymous. I think between 2005 and 2010, there wasn't a single paper that mentioned this exposome. And I have to say there's a debt of gratitude to Stephen Rappaport, actually, who picked this up in the literature and I think convinced the National Academy of Sciences to run a workshop in 2010. And I'm not saying this only because David is here, but I think NIEHS, David Balshaw and Linda Birnbaum and colleagues who promoted this idea in the US, and also the European Commission in Europe that have invested in this area. But we're in a very early phase of practice, probably five, 6 years of work where this has really been tested. So I want to give now three brief examples from some work at the agency, staying away mostly from what will come during the rest of the day. And I've picked three examples, one from the diet, one with a natural toxin, and one with smoking across these three areas. Just before I do that, I'd like to mention that some of the examples that I've seen coming in the literature of innovative ways to take this concept into practice. One of them being the public health exposome and preterm birth rates, looking at county-level variables in relation to that endpoint. There have been, of course, a number of studies on the NHANES databases looking at all-cause mortality, blood pressure. I put two examples up here. And then I thought another interesting approach was to look at pharmaceutical prescriptions database and subsequent cancer risk. So building, I guess, components of an exposome using different sources of data linked to different phenotypic outcomes. So I'll talk a little bit now about dietary biomarkers, because I think this is one area of cancer research where the methodology needs to improve if we're to really unravel some of the complex associations between diet and cancer. And so one might think of the food metabolome as a component of the exposome and translate that down into some measurable number of dietary biomarkers. And this is one of the approaches we're taking at IARC. We start with a big advantage because we have the EPIC study, so a European-wide study of diet and cancer with questionnaire data, 24-hour recall on subsets of subjects and biospecimens. So just give one example here of studying just under 500 subjects, correlating the 24-hour dietary recall data with urine samples. And we were interested in trying to develop biomarkers of polyphenol-rich foods, or a dietary component shown down here. and identified about just over 2,000 features that were correlated with these 6 different food types. And then, because we have a Phenol Explorer database, we were able to annotate a number of these compounds, these metabolites, and identified about 70 different metabolites correlated with these different dietary components, and in particular identified structurally these 6 metabolites that were the best predictors of those food intakes. I think this is one of the first examples where we were really able to start with a very broad approach and work back towards some specific dietary biomarkers. We've followed that up with some additional studies, particularly on intake of animal foods. And in this study that I show here, this is a small intervention study, just 10 subjects per group, but they were on a controlled diet, 3 weeks of increasing level of intake of different types of meat or fish, and 24-hour urines and serum samples were collected throughout that study and then analyzed by mass spec. And this is just one example of one metabolite that was already known, anserine, to be correlated with some meat intake, and you can see this particular metabolite responds very strongly to increasing amounts of intake of chicken versus the other types of meat and fish on this slide. And we're then able to go back to the EPIC study, which was a cross-sectional study, select subjects that self-reported high or low intakes of those particular types of meat, where we also had 24-hour urine samples, and test now those specific candidate biomarkers. And again, I just show for purpose of illustration anserine here, coming up very strongly compared to the other types of meat. So this confirmed really in this cross-sectional study what we'd seen in the small intervention study. And this is the work of Will Chung, who just left the agency, working with Augustin Scalbert and other colleagues. What was interesting in that study was that, as well as the metabolites that could be annotated already from the databases, there were a number of related metabolites that correlated with those, but that we don't yet know the identity of. And I try to show this on this slide. So TMAO was linked to the fish intake, but there were other unidentified metabolites linked to that. There were a group that would link very specifically to chicken consumption, only a couple of which could be identified at this stage. Carnosine really represented meat intake across the board there, and then others which included also the fish intake. So the point I'm trying to make is that we identify very specific biomarkers for some of these intakes, but others that cover a range. And there are some interesting metabolites that have dropped out of this analysis that we can now try to identify that may increase the sort of power of these dietary biomarker studies, and that's a piece of work which is ongoing. Second example I want to give is identifying new carcinogens from genomics. And this example, I think, is the first I'm aware of where the molecular biology has sent us back to the epidemiology with a new hypothesis. This was a study that was conducted, a collaborative study, which IARC was heavily involved in, looking at the genomic landscape of clear cell renal cancer. So this is the most common form of kidney cancer. And what we did was have whole genome sequencing across the kidney cancers from different populations. So under the microscope to the pathologist, all these cancers would look the same histology. But when you do the sequencing, you can see here from Romania, I think just by looking by eye, these are the different base pair substitution mutations in the tumors. And you can see that there's a very high proportion of AT to TA, the green color here, compared to the other population of tumors, or tumors from other populations. And this was repeated in a subsequent series of samples from Romania. So here we see a very striking difference at the molecular level for this tumor. What's driving this? What are the risk factors? And in fact, we had evidence already on a particular known human carcinogen, aristolochic acid, coming from this plant here, that's been shown to be linked to a different type of cancer, urothelial tract tumors, but not kidney cancer, but had very much the same spectrum of mutations. So this gave us a new hypothesis about the cause of kidney cancer. And in collaboration with Dr. Rob Tureski and his team in the US, we're able to look at the presence of aristolochic acid DNA adducts in the normal kidney DNA from those different populations of tumors. And what this panel here is showing is the correlation between the proportion of the mutations which were that signature AT to TA and the level of DNA adduct in the target tissue. And we did not see this adduct in tissue from the other three populations, only in this is just the Romanian data here. These regions, which I won't go into, of high incidence of this kidney nephropathy, this nephropathy. So in that case, I think we have a very nice biochemical support to the molecular data. And we actually now go on and see this typical mutation spectrum in numerous cancer types. So this is the upper urinary tract urothelial tumors here, the ones I've just shown you, but with additional data, bladder and some liver cancers as well. I think this is very exciting because it's taking that molecular signature and implying something about cancer etiology. I think there's a great opportunity for us to match that type of observation at the population level with experimental systems. And again, at IARC, we're trying to do this through the use of different epithelial fibroblasts, mouse epithelial fibroblasts, with or without the human P53 gene knocked into those cell systems. We can challenge and look at mutation spectra with candidate carcinogens. Also, using existing carcinogen bioassay data treated with some of these carcinogenic agents and match that to the measurement in human tumors and also what's in cancer genome databases. And that's been a fruitful exercise, at least at the moment, in proof of principle. And again, I'm not going to dwell on this, but these are spectra with benzoate from the mouse embryonic fibroblasts here, the signature in human tumors, and similar for alkylating agents, aflatoxin and aristolochic acid at the bottom. So we're seeing that the experimental models can really recapitulate what we see in human tumors. The last example I want to give is slightly different in terms of early life exposure. And here, I think it comes through this difference between using the exposome to characterize the exposures themselves and being able to also link that through to the biological consequences of exposure. And this slide just illustrating some of the interests we have in the early life exposures, even in utero stage, and that because of the availability of a number of birth cohorts, actually able through that to study the exposure and some of the biological consequences of early life exposure on the child's biology. And realize this is a very complicated slide, but we have one example up here in the top right-hand side of the slide where we had measures of maternal smoking during pregnancy, and we're able to look at the differential methylation CpG sites associated with that smoking pattern in newborns, and that really illustrated a number of biological changes associated with this exposure. So for me, the interest is not only in the specific situation, but the general principle that one can address this type of question. And, sorry. So we were able to take our studies of aflatoxin, where we've for many years been aware that exposure occurs both in utero and very early life and on throughout the life course, and look at the impact of that exposure. So Here we used our biomarker of aflatoxin exposure in a number of pregnant women, increasing levels of exposure here. And we're then looking at the differential methylation of genes across in infants aged 2 to 8 months of age. And again, I think for purposes of time, I go quite quickly here, but we're able to compare the level of exposure of aflatoxin on the individual level with the different pattern of methylation, and then identify a number of those genes, some of which were quite interestingly involved in aflatoxin detoxification process. So I'd now give a little bit of perspective, and I have to say this is just a personal view on some of the areas that we need to address if we're really to turn this concept into a useful reality in the future. And I think there are three major methodological challenges for the teenage exposome, and I'm just going to briefly go through these as a way of stimulating some discussion for the rest of the day. One is validating exposure measurements, the second is data integration and analysis, and the third is linking these measurements with the underlying biology. So I think there are a wide range of analytical challenges, and I think this audience would be very well aware of this type of problem for the exposome. You know, reliability of individual measures within a large number being made simultaneously. And that's not only actually for the biomarker area. So they may have different degrees of confidence around those several 100,000 measurements that are being made. There will be technical challenges in terms of recoveries and stabilities of the different things that we're measuring, the sensitivity, low abundance signals, how do we identify those, the reproducibility, and so on. Also, the properties of each of those individual measures, the intra-individual variation, heterogeneity between markers, and the common challenge of looking at past exposures. So the sum of all that would be variable degrees of exposure misclassification across the wide range of measurements that are being made. And there's an excellent paper online from Dennis and colleagues in EHP, which goes into some of this in more detail. In response, one of the things we were interested in doing at the agency is bringing together information in one place where at least this type of question can be considered. And Augustin Scalbert and colleagues have developed this Exposome Explorer, which is available online now. This focuses on biomarkers, principally of diet and pollution. And it gives detailed information on each of the studies that have been done. I think there are about 600 or 700 papers from which this information has been extracted, about 10,000 individual biomarker measurements. And it's also a place where he's included the correlations with external exposures, but also some of the reproducibility of the biomarkers over time. And so you can go into this database and look at things like the intra-class correlation coefficients for different markers. And as I say, particularly a lot of information on different dietary biomarkers if you're interested in that. I think this sort of synthesis of data, and I know there are other databases that are trying to do similar things, is very helpful. The relationship between the exposures that are being measured, the markers that we have, is also an important area for research if we're thinking about data integration and analysis. And I particularly draw attention to this paper by Patel and Manrai, and had a very nice talk yesterday afternoon. I think there is another in the series today on this subject. This attempt to look at the structure, the correlations between different components of the exposome to understand the co-occurring exposures. And I liked very much this suggestion that we may be able to reduce the dimension of these data through almost an equivalent of a haplotype, sort of tagging certain exposures which represent a class of exposures. But I think this is very challenging when you start to think of populations globally. So the same correlation observations may not be valid across different populations. It allows us, though, to think about potential confounders within a study, and also disease-related combinations of exposures, possible synergies between exposures. So I think this is one of the major areas of research that's needed in parallel to the development of the exposome more generally. And this is driving us, of course, towards new statistical computational methods, data mining within bioinformatics to deal with the large, complex data sets. I think there are also some technical challenges just with merging data if we want to do international studies across different projects, different platforms, and also, of course, ethical and legal challenges to overcome. And the final thing I want to mention is really the need to match the omics measurements with the biology, and this is specific to that category of biomarker. Because eventually, if we identify a phenomenon, a marker which is associated with disease, but we don't know what that marker is, we're left without any ability to modify something in order to change the disease development. I think also, and I don't have an answer to this, there's no fixed line you can draw in the sand, but I realize at times we confuse the measurement of exposure with the biological effects, the responses. And in some cases, I've seen the exposome described as the summation also of the biological responses to exposure. But at what point do we stop that continuum? I mean, would you say that a polyp in the colon is part of the exposome, or is it part of the disease? So what I would ask is that we think very carefully about the interpretation of what we're measuring. Is it exposure, or is it a normal cellular process? Is it an exposure, or is it a pattern related to exposure, or is it a response to exposure? And that's really crucial in the interpretation of the data that we will generate. Of course, there's statistical as well as biological investigation of mixtures and interactions. I think the exposome does allow us to explore mechanisms. We can look at the biological plausibility of the exposure disease association by unpicking some of the pathways that are modulated by different exposures. And Paolo Vinese has really promoted this idea of the meet-in-the-middle approach to establish causality. I think that will be discussed later in the day. So as we generate this data, we can put some structure on it, perhaps around these 10 key characteristics of human carcinogens, looking at the pathways which are modulated. And the exposome approach, I think, can start to reveal these interactions and also perhaps be mapped onto the modulation of these 10 key characteristics. And I think, given the time, I won't dwell on this, but I want to make the general point that aside from characterizing the exposure, we're also able to see how that exposure translates through to biological changes which are coherent with the disease development. To finish, then, I'd like to make a plea that we remain focused on the question. I mean, our business is in trying to characterize the exposure-disease relationship and intervene in that. So the exposome should help us frame questions, but to do that well, we need to avoid confusing the technology with the science. And I think we're at a stage of really taste and see. I mean, we now need to try to apply these approaches and see what happens, what problems we face, what challenges need to be overcome. And I'm sure that practice will drive further methodological development. And then I complete this by saying that we're at a very early stage, as I said partway through my talk, probably 5-6 years of practice in this area. And I think that requires really continued commitment in funding, including actually investment in method development and validation. And as we know, that's often a difficult area to attract funding into. But without that, the huge investments in some of the population-based studies that we're making will not come to full fruition. So my penultimate slide, I spent my summer in Normandy beautiful part of France. And I came across this memo from Churchill to Mountbatten. It says it's a very terse memo. And the problem was how to land vast amounts of equipment and men on the Normandy beaches to support the D-Day landings, subsequent to the D-Day landings. And I like this memo for two reasons. But what he said was, piers for use on beaches, that's the subject. They must float up and down with the tide. The anchor problem must be mastered. Let me have the best solution worked out. Don't argue the matter. The difficulties will argue for themselves. So I like it because that's the way that a leader should write a memo. I don't have the solutions, but I can tell you what the important question is. But also, let's not focus on the difficulties theoretically, because they will take care of themselves. What we need is to try to come up with solutions, and the solution is shown here, and really quite a remarkable feat of engineering. So I thank you for your attention. I hope that you'll get a good picture of the Exposome over the day, and happy to take questions if there's a minute or two left. Thank you.
Thank you very much, Chris, for a wonderful presentation. The presentation is now open for questions. If you have questions, please state your name and your affiliation so that people that are watching online also know who is speaking, because you won't be on camera. Paul.
Paul Schepers, Radboud UMC Nijmegen. Thank you, Chris. It was very inspiring, your talk. One thing I think that struck me that you said, it's really important, you know, to make a distinction between the health response and from what point we can actually see adverse effects. And that's a very critical, I think, remark that you made. We're trying to move away from the use of animals, as you know, to like more in vitro systems. And I think, well, of course, the experimental, the way of doing animal experiments, you know, then you can control environmental factors and have a very good, well, model to do a study of adverse effects. Do you see, how do you see that we could move away from the use of animals?
Yeah, it's. I mean, the example I gave was the mutation work, and I suppose that what we're doing is taking existing historic samples from animal experiments in collaboration with NTP and comparing those with the sort of in vitro models that have now been prepared and developed. And so we can actually ask the question whether, at least at the somatic mutation level, we can reproduce what we see in the animal studies within those in vitro systems. So I think it would really be a case of the biological endpoints that are being looked at and trying to unpick that through a case-by-case basis. But it seems to me that's a good example of where we can now say what we see, at least in the first compounds we've looked at, quite a similarity for somatic mutations. Whether that would hold for other types of biological effects, I think it really depends on the endpoint that one is looking at.
Olivier George at the University of Michigan. It's interesting to see how the Exposome has developed mostly on biomarker side. I get the impression that the predictive approaches are underused, in fact, to expand what we presently observe over lifetime, because, I mean, modeling, we can go back and do that. What is your perspective and how you see that?
So you mean to reconstruct the past?
Reconstruct the past and modeling, the role of modeling in that.
Yeah, that's an interesting point. It's not something I have a lot to say on. I think well, you just raise it, and it's very interesting. And I'm just drawn back to thinking about some of the research I was involved in with looking at liver cancer and with hepatitis B. And in fact, the strength of association between infection and liver cancer improved with the quality of the biomarker, and particularly when we had a biomarker that was an indicator of chronic exposure to that virus. So I think the point is excellent. I don't have anything to offer by way of examples on this, but I think it remains one of the major challenges, actually, particularly with the biomarkers. I think it would be a mistake, though, to put all the emphasis on using that category of marker as a way forward. I remember a very passionate speech by an Italian colleague of mine several years ago who said, you know, if the test is better than the question, use the test. But if the question is better than the test, then use the question. So we really need to, you know, for the past exposure, maybe it's at the moment the biomarkers are not necessarily the best approach.
Okay, thank you very much, Chris.
Yeah.
And due to time and being able to get everybody on time in their own sessions, I would like to thank Chris again for a wonderful talk. And of course, we hope to see you the rest of the symposium here as well. Thank you very much.